This morning, we're going to spend a couple of hours exploring several science topics of interest to the LHON community. In addition to each of the speakers, I bring to the conversation 13 years of experience with LHON from the patient and family perspective. Joining us throughout the session is Dr. Marie Denome, a neuro-ophthalmologist at the Mayo Clinic here in Phoenix. Dr. Denome will be presenting a session later this morning with Jessica Loomer and is able to provide the perspective of a neuro-ophthalmologist should questions arise during these topics. I met Dr. Denome at Nanos back in 2020. You may remember that time before we had this pandemic, and we went to conferences and she was giving a talk, and I went up to her afterwards and said, "We are having a conference about LHON in Phoenix in June. Can you join us? Can we talk about that?" And she was like, "Sure, tell me where to be. Looking forward to it." A couple of years went by and I emailed her and said, "Remember that thing we talked about a couple of years ago? Well, now we're going to do that." "Absolutely. Tell me where to be. Happy to be a help." So she's here. Could you tell the group a little bit about yourself?
Good morning. Very happy to be here. So just as Lisa said, I am a neuro-ophthalmologist. So I have the privilege of really caring for patients that have neurology-related disorders that affect their vision and visual pathways. And so a large part of my practice has to do with optic nerve disorders. So I do see Leber's patients, diagnose them many times initially, where they come to me for second opinions or so forth. My training is as an ophthalmologist, and then I did specialty training as a neuro-ophthalmologist. And then 5 years ago, my cousin's son was then diagnosed with Leber's optic neuropathy, and you'll get to meet Zach during my talk a little bit later. So I have a lot of connections with your group, both professionally and personally. So thank you for inviting me.
The topics we'll be discussing this morning are frontiers of genetics in LHON, LHON and Charles Bonnet syndrome, the LHON plus spectrum, clinical insights and perspective toward precision medicine, LHON and migraine, and LHON data collection program. Our first topic is frontiers of genetics in LHON. This talk will be given by two genetic counselors from the Mitochondrial Frontier Program at CHOP in Philadelphia. CHOP is a leader in the field of genetics and mitochondrial disease. Elizabeth McCormick spoke to us at our 2015 conference, and she's deeply involved in important work to classify mitochondrial variants. She's joined today by James Peterson, and both have experience working with LHON and other mitochondrial disease patients.
Good morning, everyone. I'm James Peterson, and I want to start off by saying thank you to the LHON community. It really is an honor and a privilege to be able to speak with you all today. And I'm joined today, as Lisa mentioned, by my colleague, Elizabeth McCormick. We're genetic counselors at the Children's Hospital of Philadelphia, and we see many patients with various genetic causes to their LHON. So for today's agenda, we're going to start by discussing genetics and LHON and identifying the specific genetic cause to a person's LHON and the features that can be seen in people with LHON. And whenever we talk about DNA, and as a mitochondrial specialist, you focus on our two genomes that we have. And so we have two pools of DNA in our body. One is called our nuclear DNA. That is the DNA that is in the nucleus of the cell. And the pool of DNA that's more relevant to most people with LHON is the mitochondrial DNA. Our mitochondria are these organelles or batteries for our body that have their own pool of DNA. And the overwhelming majority of patients with LHON have a variant or a mutation in their mitochondrial DNA, as many of you are, I'm sure, well aware, that is the cause of that, their vision loss. But taking another step back, what are mitochondria exactly, and what do they do, and where does this analogy of the battery come from? Well, our mitochondria, again, they make energy, and they make energy through this molecule called ATP. And this ATP is made through a process called oxidative phosphorylation. And the way I describe oxidative phosphorylation to our patients is that our mitochondria have a series of 5 energy factories that help produce this energy that then gets distributed throughout our body. And that's where the analogy of a battery comes from in the mitochondria. And there are other terms that are used like electron transport chain. When we talk about oxidative phosphorylation, really what the electron transport chain is it's just the first through the 4th energy factory, and oxidative phosphorylation is all 5. Now, one of the reasons why I wanted to highlight this is these energy factories. They're called complexes. And the reason they're called complexes is because they're made up of multiple different proteins that come together, and they're complex. And when we think about the genetic cause to someone's LHON, many times they have a variant or a mutation, those 2 words are synonymous, that disrupt one of the building blocks of these complexes that come together. And many of you have probably heard something similar to this, but our DNA is a string of letters that act as a blueprint to our body. They provide instructions on how our body is supposed to make certain proteins. And when someone has a variant or a mutation, that means the letter we expected to be in a certain position is not there, and it's switched out for a different letter. And we have 4 letters in our DNA, and they are A, C, T, and G. So a variant might be in a position where we expect someone to have a G, they may have an A in that position. When we think about the mitochondrial DNA, many of the genes in the mitochondrial DNA turn into a protein that goes into one of those complexes that helps our body make energy. And specifically, when we think about, oops, am I too close to the mic? Is that where the feedback's coming from? These changes, they go into one of the building blocks of the first energy complex. There are other energy complexes, such as the 5th energy complex that may have been reported, but primarily it's the first energy complex in LHON. And when we look at this map here, you'll all notice that we have the 3 common LHON mutations, the 3460 G to A mutation, the 11778 G to A mutation, and the 14484 T to C mutation. What we wanted to highlight with this map is there are other mutations as well. And I highlighted the common mutations, and I think my colors got a little bit switched here, so sorry about that. But the common mutations are the blue arrows and where they're located on our mitochondrial DNA, which is shaped like a circle. But then with the pink arrows, we see other mutations that have been reported in people with LHON, such as the 4171 G to A mutation. And there are several other mutations that cluster around the 14484 position that can also cause LHON. And what we wanted to highlight here is there are not just 3 mutations that can cause LHON. There are other mutations that can cause LHON as well. Some of you may have heard of the term haplogroup, and this was something that we wanted to address in our talk today. And what a haplogroup is it refers to the changes in our mitochondrial DNA that don't cause symptoms. And when people use the term variant or mutation, what it's important to know is we all have thousands of mutations in our mitochondrial DNA, and or I should say our DNA generally, and the majority of them don't cause symptoms. The majority of them are what we call benign. And what a haplogroup is it's the collection of variants in our mitochondrial DNA that come from our ancestral background. And if you've ever heard Doug Wallace speak, he's very elegant when he talks about haplogroups. But what we see is that as our ancestors migrated from Africa into the Middle East and into Europe and into further parts of Asia, our mitochondria adapted to different climates, to different altitudes, and they acquired mutations that allowed our ancestors to thrive in those areas. Now, those particular mutations they have don't cause symptoms, but the theory is that the constellation of those benign mutations may influence the likelihood someone develops vision loss, or it may act as a protective barrier against developing vision loss. Now, this is a growing field of knowledge, but it's something that we're keeping our eye on and tracking more so we can get a better sense of, when understanding things like penetrance and likelihood of developing things like extraocular symptoms. Does haplogroup play a role in this? And there's some evidence to suggest that it does, but we're still working on better understanding that. Whenever we talk about mitochondrial DNA, one of the most important distinctions from mitochondrial DNA and nuclear DNA is that our mitochondrial DNA has many copies, whereas our nuclear DNA is the DNA that you learned about in high school, 23 pairs of chromosomes, one from mom, one from dad. But our mitochondrial DNA, there are many copies, and when a mutation is present, there's this term called heteroplasmy or homoplasmy. And what heteroplasmy means is it refers to the percentage of copies of mitochondrial DNA that carry a particular mutation. And when we look at this little diagram here, the child on the right that is completely shaded in white, that's reflecting a child who does not carry a mutation at all. And so they would be homoplasmic for the reference. Whereas the child all the way on the left, who's shaded in blue, they would be considered homoplasmic for the mutation. And then the two children in the middle, they're what we call heteroplasmic. They have a mix of copies of mitochondrial DNA that carry a mutation and a mix of copies of mitochondrial DNA that don't carry the mutation. And so that's the difference between heteroplasmy and homoplasmy. Heteroplasmy is a mix, meaning someone might have a mutation present in 60% of their copies of mitochondrial DNA. Whereas homoplasmy, which is of course what we see most commonly in LHON, refers to someone who carries a mutation in every single copy of their mitochondrial DNA. And I was doing a little bit of foreshadowing here for my next slide. Most people with LHON carry their mutation in every single copy of their mitochondrial DNA. There are some cases where people have had it at heteroplasmic levels, but those are few and far between. And when we think about genetic testing, one of our roles as genetic counselors is evaluating the methodology that was used for genetic testing. And if you look at your reports, you may see that there's a percentage associated with the mutation. You may see that it just says that there's a mutation present. And this is a difference in the methodologies. And one of our roles as genetic counselors is assessing what are the strengths and limitations of a particular test, because some tests, again, test for the absence or the presence of a mutation, whereas other tests will get down to a level of 1.5% heteroplasmy, meaning they could detect someone who carried, just using for an example, the 11778 G to A mutation as low as 1.5%. And lastly, what I wanted to come back and review with LHON is the overwhelming majority of people who have LHON have their symptoms due to a mitochondrial DNA mutation. But what we've recently learned is that there can be nuclear DNA causes to LHON, and there's a particular gene that was recently published called DNAJC30. And the major difference between mitochondrial DNA inheritance, which we'll chat about in just a few moments, is that mitochondrial DNA is inherited from our mother. And so people who have a mitochondrial DNA mutation, and the way you know whether you have a mitochondrial DNA mutation is when you look at your report, or as your report is read to you, if it starts off with the letter M and then is followed by a period, that refers to a mutation in the mitochondrial DNA. For example, the M period 3460 G to A is a mitochondrial DNA mutation, whereas nuclear DNA mutations start with the letter C. And so I have some examples down here. So if a person had LHON due to DNAJC30, they would have two mutations that each started with DNAJC30, C period, and then there would be a string of numbers, for example, 152A to G. And so the key point there is if you see an M period, that's mitochondrial DNA. If you see a C period, that's nuclear DNA. And nuclear DNA inheritance is a little bit different because DNAJC30 is inherited in what we call an autosomal recessive manner, meaning mom carries a single mutation, dad carries a single mutation. Neither mom nor dad have symptoms of LHON, but there's a 25% chance that they pass on both mutations to their child who could have LHON. And again, this is evolving and emerging knowledge in the field of genetics. It doesn't change what we already know about mitochondrial DNA inheritance and maternal inheritance, but it just adds additional thoughts to providers when thinking about people who have symptoms of LHON about the best ways to test and get a diagnosis for those people. And at this point, I'll pass it to my colleague, Elizabeth McCormick.
Hi, everyone. I'm Elizabeth McCormick. I'm a genetic counselor at Children's Hospital of Philadelphia. And I'm going to take what Jamie had just told you and kind of bring it into the practical sense, what this means when it comes to genetic testing, what this means when it comes to those of you that have LHON and your family members as well. So when we think about genetic testing for LHON, there's many different routes that you can take. So most commonly when someone comes to our center, again, we're a genetic center, many patients come to us with testing of those 3 common variants. That's great. It's a great place to start. It can be helpful if it comes back positive. You'll know which of those common 3 variants is causing the concerns in you. However, if it's normal or negative, it's actually not too helpful because we have a lot more work to do. You could have one of the other causes of LHON, which Jamie had talked about, whether it's a different change in your mitochondrial DNA or the nuclear DNA, or you could actually have one of those common 3 mutations at a heteroplasmy level that's below what that common testing can detect, which is typically about 50% heteroplasmy. So the next step in comprehensive testing for LHON would be sequencing all of your mitochondrial DNA. So not just going in looking for those 3 common typos or mutations that we can see, but sequencing that whole, all of the letters in the mitochondrial DNA. And what's nice about that is many of the labs that are doing that now can tell you if the mutation is there or not, but also what the heteroplasmy level is. And then you can also get information on the haplogroup, which Jamie had told you a little bit about. But again, hopefully you all know now that there are also some nuclear DNA causes of LHON, as well as optic atrophy and other optic nerve concerns. So if you had testing for those common 3 variants and mitochondrial DNA testing, and it's still normal, you still don't have the answer, I would really encourage you to keep looking because as you probably all know by now, LHON and mitochondrial disease doesn't always follow the rules, even their own rules sometimes. So it's really important to take a very comprehensive look. So you can have exome sequencing that actually sequences all of your genes in your nuclear DNA. It's not all of your genetic information. The genes are the part of the genetic information that code for something, but exome sequencing wouldn't include those regulatory regions that turn genes on and off and up and down. That's whole genome sequencing. And those tests are becoming more and more available. So I guess really what I just wanted to make sure was clear here, if there's any of you in the audience that don't have a genetic diagnosis yet, it doesn't mean that your vision problems aren't caused by a genetic change. It probably more or less means that we just haven't found it yet. We have many patients in our center that have features classic for LHON that we haven't been able to find the genetic cause. But it's important to know we have 20,000 genes and we really only know what like 5,000 to 7,000 of them do currently. So that's not to say those other 13,000 genes aren't important. We just still have a lot more work to do on the science side to figure out exactly what those genes do and how they might be related to medical concerns in people. So we just wanted to show this picture again for heteroplasmy. So again, now we're back to thinking strictly about mitochondrial DNA changes. But why this is important is because thinking now more about family members, you can sometimes have family members that have different levels of this variant at different levels of heteroplasmy, even within the same person and different parts of them, whether it's blood or skin or a cheek swab, you can have different levels of these mtDNA changes or pathogenic variants. So it's important to know, just because you have information on maybe one member of the family or in one tissue in one person, you can always do additional testing to get, a more comprehensive picture of what those heteroplasmy levels are. Typically, when it's homoplasmic in one person or in one family member, it tends to be homoplasmic in other parts of that person and other family members. But as I said, there's exceptions to every rule. So to continue along those lines, some people in a family that have those LHON common variants and even the less common causes can have different medical concerns. So I know you're going to hear later about the other organs that can become involved other than the optic nerve. But again, in a family, I know it sounds crazy, you can have a family member that has the same exact genetic cause. Those two people can be affected differently. Because remember, these are just one gene and one change among 20,000. So everyone's different, even among the same family. So just wanted to keep in mind that just because one person maybe only has optic nerve problems, the other family members could very well have a different presentation of their medical concerns. So again, I won't spend too much time on this because you're going to be hearing about it later. But some of the other medical concerns that like we look for in family members that come to us with an LHON variant are multiple sclerosis-like symptoms. They can have other nerves, so not the optic nerve, but other nerves in their body, problems there, different muscle problems, hearing concerns, whether that's a sensory neural hearing loss involvement, so not the optic nerve, but the nerve that goes into your ear, as well as we understand there is a very high prevalence of some processing changes when it comes to hearing and auditory concerns. There have been some reports of cardiac involvement. It's not exactly clear what the chances of that cardiac involvement is, but it's also relatively easy to go to the cardiologist and get that screened. So again, that's something you might hear about later today. And again, it's one of the things that's really perplexing to us is that it's really not clear, as I was saying, why some members of the family have certain organs involved and others maybe have more involvement. So we think it's genetic. I'm biased because I'm a genetic counselor. I think everything's genetic, but it's just for us to keep trying to figure out. So just to talk briefly about family members, when it comes to the chances that you could or could not have vision loss associated with these variants. So I will say these definitely are the classic numbers. Groups you might have heard on Wednesday are doing more work to really fine or tune the chances that someone may or may not develop vision loss and the other medical concerns associated with these variants. But not only is the organ system involved variable, the age is very variable. We have some patients in our center that had vision problems early in life, and then we have some adults that didn't really have any changes until they were older. Yep. So that's coming to the end of what Jamie and I had wanted to talk about. So again, really the take-home message, if you had genetic testing done, it's not quite clear to you, is this the answer for you? Is it not? Do you have more testing that needs to be done? Always ask. It's complicated, even for us sometimes, things change and we get thrown a curveball here and there. So it's okay to kind of ask for additional guidance and make sure that it's crystal clear to you what the genetic cause for your condition is, and then what that means for other family members as well. And again, the other take-home message, if there are any of you out there that don't have a diagnosis, it doesn't mean there's not a genetic cause. It just means either you need more comprehensive testing and we need to keep looking for that answer. And then again, the symptoms are very variable. So as I said, mito disease doesn't always follow the rules. So sometimes we have to do a little bit more digging. I think that's it.
Thank you so much, both of you, James and Elizabeth. We do have a few minutes for questions. We do have folks with microphones. And if anyone does have questions, Of course, it's always in the back of the room that the first question comes, has to get the mic runner running. If someone else has a question and you want to put your hand up now so we can be prepared, that would be awesome.
Okay, sorry. So are there distinct triggers for the two variants of LHON DNA? Does that make sense? Or is this the right word to ask about? So you said that there are two different versions. You can have the mitochondrial DNA or not. And my question is, there's a certain list of triggers for vision loss with, so are they the same?
Yeah, so typically the triggers that you're thinking of are more known for the mitochondrial DNA causes. We're really just learning about the nuclear DNA causes. So I don't know if anyone has done enough or looked at enough patients with those causes to really know. So it's hard to know right now.
Thank you. I have one more question. In terms of LHON plus, I just want to, my understanding is you can have LHON plus without vision impairment.
Can you hear me? Okay, perfect. That's an excellent question. And I think this is something that is debated in the community of when does LHON set in? Is it necessarily at vision loss, or is it when people start experiencing their first symptoms? And I wish I was able to give you a more direct answer. I think what would happen is if you asked 10 different clinicians, you might get 10 different responses. Our approach is that when we see someone who carries a pathogenic variant or mutation, we review their review of systems, and we see what symptoms they have, and we review the literature then for that particular variant, and we provide care based on the symptoms that overlap with what's been reported in the literature. I say that knowing that if you were to ask another clinician, they may say that when LHON starts is when the vision loss starts.
Yeah, and just to further that a little bit, so this is something we've talked about and we've seen. So when I guess Dr. Lieber named LHON, we didn't really know the genetic causes at that point, right? So he saw set symptoms in a set amount of people and called that LHON. But now we know with the genetic causes, everything is a spectrum, right? You can be less severely affected to more severely affected. So if you have one of the variants that can cause LHON, but don't have the vision loss, well, you still have that variant, but I guess you don't have the vision loss. So it kind of depends on, what you consider LHON. Again, I'm biased, I'm in genetics. So I think the genetic diagnosis is the most important. And then what you call the features is what Jamie was saying is can be variable from doctor to doctor.
Okay, so my last question. I'm really sorry.
Great question.
I guess my question is, and maybe I should ask for a clinician who treats people with LHON or LHON plus. Is there a reason really to pursue getting an LHON plus diagnosis if you already know that you carry your period?
So in our center, we are fairly cautious. So if we have someone that has one of the variants and they have the vision loss associated with LHON, we just send them to screen for those other organ system involvement. It's easy for me to say, but it's relatively easy to have the heart checked out, the ears, maybe have a comprehensive neurologic evaluation. Because if something's there that's potentially treatable, well, why not find it early? If you go to those specialists and everything's normal, well, that's great too. But it's good to know if there is something else there to see if there's something that can be treated.
I just wanted to mention, follow up on Robin's question, which caused me the question. As you say, individuals are curious and they have that mutation, then overall in your consideration, they have LHON. And so having said that, we're not under socializing this in the UK, Australia, working on the bus and us. It's really not good insurance. And those of us who are blessed to have good insurance and could do those baseline tests. Sorry to the hearing, etc. We just mentioned as carriers, would you recommend it as carriers and not just for those who have already experienced vision loss?
That's an excellent question. And I think the one point that I would amend at the beginning of your statement is if you don't have any symptoms currently, we would say you are at risk for developing LHON. But the second half of your question there, we do screen unaffected carriers with, again, ophthalmology evaluation. And I'll let Dr. Denome go into the details. I won't presume to understand the specific anatomical changes that are seen, but I do know that unaffected carriers can have changes on the back of their eye without the visual loss symptoms. But our center is recommending screening eyes, ears, heart for currently unaffected asymptomatic carriers, just as a baseline screen, and going from there, and then keeping in close touch with their primary care provider if they experience any change in their baseline status.
Excellent. Thank you. Any more questions? I see one coming.
Curious, about hearing loss. Because not as effective when I was 10 years old, 15 years old, and that's significant hearing loss in my right ear. Never no idea loss.
So there's no way to know definitely, and hearing loss can be common as you age, so it can be hard to know. Typically, when we think of hearing loss in mito disease, it's typically in both ears, typically bilateral. So anything that's one-sided, we always a little bit less suspicious.
Got one more?
One more. Is there any significance to the alpha characteristics after the their portion of the
Like the A to G.
That's a great question. And so after the number, the two letters that are presented, the first letter is what we call the reference, meaning that is the letter we're supposed to see in that position. For example, 11778 in the 11,778th position, there is supposed to be a G in the mitochondrial DNA, and people who have the common mutation have an A in place of that G, and that's what that G arrow A means. And the first is the reference, what letter is supposed to be there, and the second is what we see in this person instead of the reference. That's an excellent question.
One more.
What's that some of the mutations mutation around
That's probably like the $5 million question, $10 million question now. Still trying to figure that out.
Just curious for the preview, to what extent beyond, to what extent have we seen some of the environmental factors impacts the diagnosis of those who we might already have as carriers, and then the impact and what are those baselines?
It's a hard one.
That's a really good question. And I wish I had a more direct answer. It's tough to prove environmental exposures as triggers or causatives, but we're seeing obviously trends in that direction across long history. And so I think knowing for sure why some people may be exposed to a trigger, you might get two people in the same family who are exposed to the same trigger. One may develop vision loss, the other may not. I don't think we understand why that happens. And to go back to Elizabeth's original point, we have over 20,000 genes. What likely plays part of a role is there are other gene variants that we have in our body that we don't know about yet that provide either added protection or added susceptibility. And that's what we're not understanding when we see the differences across different family members. Is again, when we think about mitochondrial DNA, that's 37 genes. Our nuclear DNA, there's 20,000 genes, and we know what less than half of those are doing. And there are millions of variants in there that could contribute a small portion of protective factor to one person in the family who carries that variant where their sibling maybe didn't inherit that variant, and so they don't have that added layer of protection. But to be completely candid, that's a bit of speculation there. So I don't know for sure.
And I think if you look at a lot of the earlier literature before we knew a lot of more about the genetic makeup, we sort of hung our hat a little bit more on that environmental trigger. But once again, you couldn't really see it in every family member or so forth. So now we lean much more heavily on the genes.
Next, we're going to talk about Charles-Bonnet syndrome. And I'm joined by Molly Garone and Paul Laurie, who spoke so effectively at the Voice of the Patient session for us on Thursday. While none of us are experts on the topic, we all have an interest in it, and we want to share with you what we've been learning about it. As I mentioned during the community update session, Molly is our education director. Outside of the LHON community, Molly works at the Prevention Research Center at the Harvard School of Public Health to translate and communicate about research to a variety of audiences. She also holds a master's degree in health communication, so her education and training make her a good fit for this role in our community. Molly will start us off by providing an introduction to Charles-Bonnet syndrome.
Thanks, Lisa, and good morning, everyone. It has been such a pleasure to serve in the role of education director for the LHON project over the past year. It's really neat to look closely at medical and non-medical topics in the world of LHON and help to educate the community and disseminate valuable information in an accessible format as best as I can. One of these topics is Charles-Bonnet syndrome, and I'm going to set the stage a bit on what it is first and its connection to LHON. So Charles-Bonnet syndrome is the experience of phantom images or hallucinations in people living with some form of acquired vision loss, such as LHON, that coexist with their regular visual experience. The cause of Charles-Bonnet syndrome is unknown, but it's thought to be the brain's response to the deterioration of vision. And on the screen, we have an image of a typical presentation of Charles-Bonnet syndrome in someone, perhaps seeing certain things in their regular visual experience, but then some unusual visual perceptions in the middle. So how does Charles-Bonnet syndrome manifest in people affected with LHON vision loss? Phantom images or hallucinations may be very simple. They can be very complex. They can be static or moving, maybe appear in color or black and white. There's lots of variation on what folks might see in their experience of Charles-Bonnet syndrome. Sometimes it can be unusual visual perceptions, such as flashes of light. And other times they can be very clear, much clearer maybe than what one's vision currently allows, or even clear enough to look real. And so on the slide right now, we have an image that is a rendering that we put together based on the visual experience of someone with both LHON vision loss and Charles-Bonnet syndrome. So it's an image of a sort of a seaside seascape, and it's blurred, of course, from LHON vision loss, but in the central field of the vision, there are some many pink dots and green squiggles, and that is an example of something that someone with both LHON and Charles-Bonnet syndrome might experience. So what does the research say about this? So there is some research on the experiences of those with a variety of eye diseases who also experience Charles-Bonnet syndrome. There's even some research on Charles-Bonnet syndrome and those who have diseases that cause central vision loss, like LHON. But in the world of LHON, there's limited research on a few cases of people affected with both LHON vision loss and Charles-Bonnet syndrome. And so now I'm going to pass it back to Lisa to talk about some of the emerging research going on in our community on these two topics.
Over the many years that I've been involved with LHON, the topic of Charles-Bonnet syndrome has come up from time to time. During one of the science team's monthly meetings a few years ago, we discussed the article shown on this slide by a team of researchers in Germany who'd studied a small group of LHON patients who reported having Charles-Bonnet syndrome. That discussion sparked a conversation with Dr. Rustam Kranja, one of the leaders in LHON research, about the need for additional research with a much larger sample size. He and his team in Ottawa created a survey about LHON and Charles-Bonnet syndrome that many of you and others in the LHON community participated in. The results were recently presented at ARVO, the Association for Research in Vision and Ophthalmology's annual meeting. Here's what they studied and what they found. They utilized a validated French-Canadian questionnaire designed to assess for Charles-Bonnet syndrome and adapted it to screen patients affected by LHON. The original questionnaire contains a total of 55 questions, 13 of which serve to detect Charles-Bonnet syndrome through a screening algorithm, while the remaining questions examine different dimensions of the hallucinations. This validated questionnaire was then modified and tailored specifically towards screening and characterizing Charles-Bonnet syndrome in LHON patients. The changes made revolved around adding additional questions designed to categorize patient age and gender, in addition to their LHON gene mutation and disease duration. There was also a question added to specifically ask if patients had ever been formally diagnosed with Charles-Bonnet syndrome by a healthcare professional. The final questionnaire was 59 questions long, organized into 3 parts to cover 9 dimensions, such as the psychological impact, context of the hallucinations, psychosocial support, and more. An electronic version of the questionnaire in English and French was created using Microsoft Forms and made available online. Information about the study and questionnaire was shared with the LHON community via email and on social media platforms, including Facebook. After excluding responses due to being invalid or by patients who indicated they were not actually diagnosed with LHON, there was a total study population of 169 LHON patients. After undergoing the survey's 13 screening questions, 118 patients, 70%, were identified as screening positive for Charles-Bonnet syndrome. These 118 patients were included for further analyses. 88% of these participants were 65 years or younger at the time they completed the survey, and 58% were male. The most common types of images seen by the Charles-Bonnet syndrome positive LHON patients were simple images like dots and spots, pulses and scintillating lights, such as flashes of light, shapes or lines or circles. A significant number of patients reported seeing more complex images like scenery, tapestry-like pattern, people, trees, animals, or faces. Over 50% reported the visual perceptions were clearer than what their vision allowed for, and over 60% reported they were clear enough to look real. LHON patients described their Charles-Bonnet syndrome experience with a number of negative outcomes affecting one or more daily activities and physical and mental health. 70% reported the hallucinations being bothersome, 55% worrisome, and 46% were scary. 36% reported that the images disturbed their sleep, and 48% reported that the images negatively affect their mood. The hallucinations experienced can appear as frequently as every day or as little as just a few times a year, and the duration can vary, lasting anywhere from just a few seconds to even being present constantly. 77% of patients reported experiencing the visual hallucinations for at least a year. Among the CBS positive patients, only half reported that they had heard of it before, while only 42% had ever talked about their hallucinations with a healthcare professional. Only 11% reported ever having been diagnosed with Charles-Bonnet syndrome by a healthcare professional. As previously mentioned, the 118 patients that screened positive for CBS in the study population included all LHON variants, including patients that didn't know which variant they were affected with, and variants other than the most common 11778, 1444, and 3460 variants. However, the results were similar if the group was limited to the 3 primary variants. The sample size was too small to do subgroup analyses. For example, they were unable to distinguish if the findings were different between males or females. This was an online survey that has certain inherent limitations, including ascertainment bias, availability in only English and French, and access to online resources. Despite the limitations, this is the largest study to date looking at Charles-Bonnet syndrome in LHON and advances our knowledge on this topic. This study leads to several important conclusions. First, Charles-Bonnet syndrome may be much more common among LHON patients than previously suspected. Second, only a minority of LHON patients are being screened or educated on Charles-Bonnet syndrome, leading to an inadequacy in the diagnosis and provision of care for this condition. Third, Charles-Bonnet syndrome can lead to various negative health outcomes. Finally, patients affected by Charles-Bonnet syndrome have interventional needs. The response data collected from this survey in itself can go a long way towards counseling patients who are experiencing Charles-Bonnet syndrome. First off, patients can be reassured by the finding that Charles-Bonnet syndrome is very common, while also explaining the variety of different ways that Charles-Bonnet syndrome can present. Establishing the diagnosis and counseling patients about it is important to alleviate stress and anxiety in these patients and to develop strategies to deal with it. These results will soon be published. Having this information available is already raising awareness of Charles-Bonnet syndrome and LHON among vision care professionals. Aside from that process, what's available for those in the LHON community impacted by Charles-Bonnet syndrome? Just knowing what Charles-Bonnet syndrome is and that it can be totally normal for those with LHON can be helpful. What treatments are available? No specific treatments, but a range of options and techniques are available to help manage Charles-Bonnet syndrome. People can seek low vision services and experiment with behavioral changes. In addition, through this process, we've learned more about Charles-Bonnet syndrome resources. CharlesBonnetSyndrome.org is a valuable resource. Vision Aware has a page on Charles-Bonnet syndrome, and the link is on the screen and is available in the next document that I want to mention, which is we have collectively developed a Charles-Bonnet handout that is accessible in the app for the conference here today in the family syllabus section. The file name is called LHON Saturday Science CBS, and we'll soon add that to the LHON.org website. The research has led to the development of relationships between global Charles-Bonnet syndrome organizations and our LHON community. Paul Laurie has volunteered to be a Charles-Bonnet syndrome ambassador in our LHON community. Paul, can you tell us about the Charles-Bonnet syndrome support community?
Yeah, well, first I'd like to say that the website, the Charles-Bonnet Syndrome Foundation website, is really, I think, an empowering and important part of understanding what Charles-Bonnet syndrome is all about. I think it's well designed. It's very accessible, and I think it's extremely informative. And I know that, and I want to thank Lissa for this. She brought me into the conversation with the director of the Charles-Bonnet Syndrome Foundation of Australia, and Scott Mearden. And he was just a really pleasurable person to talk to, and is really passionate about Charles-Bonnet syndrome across the entire spectrum of low vision and visual impairment conditions. But I think it's really important, if you are, dealing with any issues related to Charles-Bonnet syndrome, to gain knowledge, and that potent knowledge can really, I think, help you overcome some of the difficulties you might be having with it. There are excellent sections of the website that are good for gaining support services and support ideas and treatment idea. Well, no treatment ideas, but really just ideas to cope. That's the better word, with Charles-Bonnet syndrome. And then I'm certainly willing to, do some one-on-one, outreach to anybody that you might know or yourself that might be dealing with it. But I would really, really encourage those of us in the LHON community to perhaps provide some feedback to any of us about what you might have done to cope or overcome some of the experiences that you've had with Charles-Bonnet syndrome, so that I can or others can, relay them to those in the community who might be, struggling with Charles Bonnet syndrome. The last point I'd like to say again, thank you to both Molly and Lissa for allowing me to contribute to the preparation for the presentation and also the preparation for the document that Lissa referred to, that about the information document about Charles Bonnet syndrome. Thanks.
So we have a few minutes to take questions. If anybody does have any questions on the topic of Charles Bonnet syndrome, feel free to raise your hand and our mic runners will get the information out. And if it's something you'd rather discuss privately, we can also during the break at 10 o'clock. We'll all be happy to talk about it then or any other time. So if anyone does have a question, raise your hand.
So while questions are maybe surfacing, just for those of you who may be thinking, so Charles Bonnet is sort of, as you think about what you may have heard of as phantom limb syndrome. So when somebody has unfortunately lost a limb, they sometimes sense or believe that their limb is still there. So this is frightening, and you're not alone as a labor's hereditary optic neuropathy patient sensing this, but we see it in our other visually impaired patients. We're still trying to get the word out. I'm still trying to get the word out to my colleagues, because in our literature, we usually talk about this occurring in macular degeneration and glaucoma patients. And so therefore, even among professionals thinking that perhaps we don't see it in other visually impaired patients, but we do. And so it's a very important conversation, because patients are frightened to bring this up to their loved ones and to their healthcare professionals, because they don't want to be perceived as being delusional as opposed to seeing just visual hallucinations, and being told that this is just a sensory phenomenon that they're experiencing as part of their eye disease. And so one, providing that comfort, and also providing that comfort to their family members as well. And then, as Paul said, trying to give them some behavioral techniques. I recently saw a woman in my office in her 60s who was experiencing frightening visual hallucinations in her field of visual loss. And it would always occur when she was looking out her kitchen window. Maybe it was the way that, I think the way the glare and the light would come in, that maybe it produced that phenomenon. And so giving her some behavioral techniques to maybe get herself out of that environment or so forth, to try to diminish the hallucination that was occurring. So definitely sharing this, talking about it, even if you're not experiencing it yourself. Once again, sharing these stories are empowering, just like I need to bring it up to my patients and to my colleagues, so that even if they're not experiencing it, they're aware of it.
Everything that Dr. Denomi just said, I didn't do. So please, heed her advice, especially, and it's probably the first thing I say in the two experiences I've had with some one-on-one contact with people in our community who have been struggling recently with Charles Bonnet syndrome. Please have the discussion with your neuro-ophthalmologist or with your healthcare team. Don't let it just, oh yeah, that's a thing, that, that's you need to have more of a conversation with your healthcare team than, yeah, that's a thing.
Hi, I have a variety of questions, but I feel like I'm just going to ask them and then just sort of what you think. And they were applying triggers for Charles Bonnet syndrome, and then triggers like having it, and then maybe triggers once you know you have it. If there are anything that triggers the episodes once you know you have Charles Bonnet syndrome, and then if there's something that maybe exacerbates those so that they're worse or not during this experience. So you kind of alluded to that just now. And then Are there also, like we just talked about, LHON plus without vision loss? Is there, I mean, I guess this would just be Charles-Bonnet syndrome.
So that's a lot of questions, but I'll try to address them. So for some patients with Charles-Bonnet, so in a visually impaired patient, they may just have sort of that phantom limb syndrome. There may be absolutely no trigger at all, except the trigger is that they have visual impairment. And so there is that sensory phenomenon that occurs. For some, it seems to be, when you listen to the patient or to the client, that for some reason, when they're in a darkly lit environment or a more brightly lit environment, or if there is waves of light coming in, that perhaps it seems to produce the hallucination. I've had some patients where it's always in a particular spot in their home that all of a sudden then they start to see these images. So I can't tell you perhaps that trigger. Now, I will tell you as a provider, though, trying to sort out, of course, we always have to sort out that there's not any other neurologic component that is there, especially in our older patients that have macular degeneration or so forth. Now, getting to the LHON plus, we'll talk about this at the next symposia, but also there can be separating out that patients don't just have a migraine aura without headache and are not seeing a sensory phenomenon, because even though you are poorly sighted, you can still have the sensory cortical phenomenon of the migraine that is there. So I hope that addressed some of your questions.
One other point I wanted to make is that something that I learned in talking with Lisa and I talking with Scott Muirden and some information on the CBS website was that certain prescription drugs can exacerbate. So certain classifications of prescription drugs can exacerbate, possibly exacerbate the experience that one might have with Charles-Bonnet syndrome. And it was from a personal experience. I use Bromidine. Is that the right name of the Bromidine drops in my eyes for eye pressure. And I didn't know, and nor was it ever discussed with me by my healthcare team, that using those drops can make the experiences of Charles-Bonnet more frequent or more exacerbated.
Our next topic is we're going to talk about the LHON plus spectrum, and we are joined by Dr. Andrea Grootman and Dr. Ann Caramello, both at Children's National Medical Center, and I will let them take it away.
So happy to be here and talk about these things, and just a kind of personal disclosure. So my mom actually had macular degeneration from a rare genetic condition, and so she did have Charles-Bonnet, but back then, they didn't really know much about it. And, I know how debilitating it was, and how when she lost her vision and couldn't drive, she lost her independence. So, growing up, I was sort of her eyes everywhere. And so, I really respect the fact that you're able to talk candidly about this, and, certainly let your healthcare professionals know, because it is a big deal. So with that, I'm going to talk about LHON, LHON plus, and LHON MS. And there's a lot of confusion in terms of the semantics of that. So just to try to break it down a little bit in finer detail, why do we need to know that? Because it is an evolving spectrum of clinical symptoms that we're learning more and more about. So if you have just vision loss, we tend to refer to that as LHON or LHON. Multiple symptoms with or without vision loss along the spectrum, LHON plus and LHON MS. So Leber's hereditary optic neuropathy is maternally inherited form of blindness due to a mutation in one of several genes in the mitochondrial genome. And although it predominantly affects males, we're now understanding that there's a broader spectrum of female involvement. And so the mitochondrial mutation can be silent until there's a trigger. And so we would consider those people to be initially asymptomatic, or there can be, once the trigger causes vision loss, then those people would be considered symptomatic. And after the onset of vision loss, most patients will experience progressive worsening within months. There are several clinical trials for this disorder. So in distinction to some of the other mitochondrial disorders, things are actually a little bit further ahead with LHON for clinical trials. And idebenone is often prescribed. That's a synthetic coenzyme Q10. And also the FB743 analog has been shown to be effective in some patients as well. So again, these are progressive disorders. So we refer to them as neurodegeneration. So the optic nerve is actually a part of the brain. It's the ganglion cells are actually neurons. And so there can be also additional extraocular symptoms with or without vision loss in this continuum spectrum. And we refer to them as LHON plus or LHON MS, predominantly affecting women. A large portion of patients will have visual impairment persisting in one eye for an unusually long period of time. It can be up to 2 years before both eyes are affected in the LHON plus and LHON MS. And also patients can exhibit other symptoms, which can vary among patients. So not every patient necessarily has the same symptoms, and it can be throughout the progression of the disease. So they're not necessarily clustered in the beginning. When the vision loss comes, they can pretty much occur at any point. We ourselves with Alyssa have done a survey. This was de-identified information. So thanks to those who participated using a digital platform on the LHON Plus Global Project, just to see, how is this distributed demographically? So as you see, women make up the majority, and older age is when we tend to see the symptoms. So although there can be symptoms in the very young children, it tends, at least in our population, to be predominantly affecting those over age 22. And then, curiously, most participants with LHON Plus had at least one family member with LHON, as you can see here. So they were asked yes or no. So about 63 or 65% had a family member with. However, if you asked another question, how many of you had a family member with MS? It was the opposite. So very few actually had a family member with MS. And so what are some of those extraocular symptoms? And so many of you know these firsthand: neuropathy, the myopathy, muscle problems, tremors, Parkinsonism, dystonia or movement disorders, bladder spasm, or other types of urinary dysfunction, gastrointestinal dysmotility, anxiety, migraines, or other types of severe headaches, and cardiomyopathy. And so the clinical manifestations are very highly heterogeneous, as you can see here. So vision loss, predominant symptom, obviously, muscle weakness, hand tremors, but seizures, in contradistinction to other types of mitochondrial disorders, very rare. And bladder spasm as well, very, compared to other symptoms, common, but not as common. And so most patients with LHON plus get a LHON diagnosis. Okay, so when we asked you, how many of you were diagnosed with LHON? Almost 70%, LHON plus about 25%, and LHON MS, very few. Most LHON plus patients were diagnosed by a combination of ophthalmologic evaluation, brain MRI, and blood testing. So the blood testing to identify the genetic mutations, the brain MRI to rule in or out MS type features, and then you've heard about all the ophthalmologic evaluation. So the majority, many have had brain MRI, subsequent genetic testing. Muscle biopsy and not really necessarily a test that we use to diagnose this condition. So although some of the participants had a muscle biopsy, it's not really something that we would jump to. And skin biopsy for diagnosis, not really used, but Dr. Caramello will talk about skin biopsy and research implications. And so the extreme heterogeneity of the phenotype or the clinical manifestations is what makes the diagnostic challenge in this disorder. So the disease progression is variable amongst patients. So I guess I don't want to overuse the cliche, but if you see one patient with LHON plus, you've seen one patient with LHON plus because they're all different. Extraocular symptoms can vary even within the same family, amongst family members. We're learning about environmental modifiers to the symptoms and diet. Diet may be playing a very important role. There are really unreliable diagnostic algorithms at this point, and the absence of disease-specific biomarkers. There are only 2 non-specific blood biomarkers that are available to denote mitochondrial dysfunction, and that's the fibroblast growth factor 21 and the GDF15. In the absence of curative treatment, patients are often put on the mitochondrial cocktail. And so depending on your provider, what is in the cocktail may vary. It may include CoQ10 or adebenone, which is a synthetic CoQ10. And that tends to have the most efficacy on vision loss. Carnitine, riboflavin or vitamin B1, B2, thiamine or vitamin B1, lipoic acid, vitamin C or vitamin K. And then so I'm going to pass it on to my colleague. And I apologize, I have to leave to go back over to the Leigh syndrome. They have me ping-ponging today, but thanks so much for your attention.
Thank you so much for joining us. As she said, we grabbed her away from her other session to come in, give a little talk, but now her colleague, Dr. Caramello, will give us a little bit more time because she always gives us so much time. So
thank you, everyone. Can you hear me? Is it on? Yeah, okay. So first I want to thank Lisa. I've been working with Lisa and Lisa, who is in here for many, many years. And I really applaud their leadership and to bring forward this LHON project. And not only they brought awareness, but they brought it to a national and international platform. So I think that deserves all kind of credits. So I just want to expand from what James and Elizabeth said earlier, but bringing at a much higher level, the cellular level. And that's the rest of the presentation will be to really try to address some of the questions, especially several questions in the back here that really went after why this clinical heterogeneity, why this genetic heterogeneity, and why do we have so many variables among patients. So it's a mystery box. I'm not pretending I just can solve the mystery, but I'm just going to present some clues that we have. So to recapitulate what James said, that no, that's not what I want. Listen. So a cell, any cell that we have in the organism, in the body, have many, many mitochondria. So a cell has not just one mitochondrion, but multiple mitochondrions. So that's why you end up having a mixed population. And then a mitochondrion by itself is just semi-autonomous, meaning that it has its own genome, the mitochondrial DNA genome that James went over. So I'm not going to go over that, but it needs a helper. And the helper is what we call the nuclear genome located in your chromosomes. And together they form that organelle. But since the mitochondrial genome is so tiny, it only encodes 13 proteins, it needs a lot of help from the nuclear genome, the chromosomes, because they bring the majority of the cells, the proteins, and that's how you make a mitochondria. So that's with that in mind, I'm just going to go over some very basic stuff such that we're all on the same page. And mitochondria are ubiquitous. They are found in all organs of the body. There's no exceptions here. There are some organs that have more mitochondria than others, depending on the level of energy they require to function. So this is, I'm missing my brain. Well, not mine, but on the picture. The heart. Okay. Thank God. I put some label here. I don't know what happened there. Okay. So you can see that the brain, you would see that there's the brain, the heart, and the skeletal muscles, the kidneys. The kidneys are very rich in mitochondria. And then the pancreas, where you make insulin. So that might explain why some patients are diabetic, because their pancreas are not working well. The bones, the intestines. And that I really want to emphasize on that because it's not really well addressed. And a lot of patients complain of gastrointestinal dysmotility. They oscillate between constipation and diarrhea. It's kind of like neglected. And that's because there's a ton of mitochondria in the intestines. And the intestine, in order to work, needs mitochondria. The eye, so that's obvious for these very educated audience, the liver, and so forth. So what they all have is that all those the mitochondria in those organs make ATP. So I just want us to demystify, why do we need the mitochondria to make ATP? Why can't we just make ATP from somewhere else? Because after all, in nature, you always have a backup plan. So how come this backup plan is not working? So what is a backup plan? So I kind of like broken down into very simple steps, and just to simplify the concept is that first you have like glycolysis. What is it? So, oops. So glycolysis is a breakdown of sugar. So when you break it down, you have 2 molecules of ATP. So it's more than nothing, but it's not enough. So not only you make 2 molecules of ATP, but you make that compound that called pyruvate that's going to migrate into the mitochondria. And that migration is essential because then this pyruvate is going to be converted to something called acetyl-CoA that's going to trigger a wheel, like a motor wheel. That's called the Krebs cycle. That Krebs cycle, every time it churns, it's going to make some products. So the output is another 2 molecules of ATP and some product that we call reducing agent, NADH and FADH2. They are going to provide electrons to trigger the making of ATP in a mitochondria. So once the pyruvate gets in, the Krebs cycle is in the mitochondria, and then it feeds the oxidase pathway. So you have just to see the Krebs cycle as a motor. Not that I'm really mechanical, but I mean, I'm pretending here. So the Krebs cycle is a motor, and then the oxidase are the wheels. And the oxidase pathway is depicted here, and it's like a little bit more what James presented. But you can see that the yellow circle or the subunit encoded by the mitochondria. So the mitochondria doesn't encode so many units of those big complexes, but enough just to create havoc once you have a mutation in one of those. And the goal here is for the electrons to come here and to hop around with a little shuttle. So the first shuttle is a coenzyme Q10. That's why, I mean, we prescribe coenzyme Q10 to patients with mitochondrial disease. And then it goes to the next complex, complex 3, and then they hop on another shuttle called cytochrome C, and eventually reach the complex 4, and that's going to communicate, provide energy, mechanical energy to complex 5 to make ATP. And at the end of this process, you make 32 to 36 molecules of ATP. This is why you need mitochondria, because you can see, you can do the math. In mitochondria, we got like maximum 38 molecules of ATP versus 2 molecules of ATP from breaking down sugar. So you can see why you have like such a deficit. So then let's go over briefly, because I mean, I think that a lot of people went over those different mutations, but this is on the left, you have the result of the survey we did with Lisa and Lisa. And we realized, we asked the patients, I mean, with which mutation they were diagnosed with. Some of them were, we were surprised, but sadly so, a lot of patients were not genetically diagnosed. So I think this is something that has to be changed, because then if you don't have a genetic diagnosis, you have like a pseudo-clinical diagnosis. So you can see the most frequent mutation is 11778. And then after that, that maps in an ND4 subunit. So we see when they're put in the red circle, Those are the mutation with the mutation. And then the next one is, oh my gosh, I even don't read myself here. I mean, oh yeah, wow, it's. Okay, the next mutation is the 14484, 13%, and then the 3460, 88%. So, and then we have a bunch of least frequent one. And so I decided just to emphasize on those, because if you're a patient with genetically diagnosed with this mutation, then you kind of are nearly all by yourself, because such a low frequency that it just doesn't generate enough communication and dialogue. So I really want just to emphasize that it doesn't matter which mutation you have. I mean, what matters, because when you have it, it's 100% for you. So then we did like a correlation between the age of onset and the mitochondrial variant. And like Dr. Gorman presented the previous graph saying, like, most of the patients, they become symptomatic 18 plus, or she said 22, but I guess I'm going to assume like 18. But we found 3 mutations that are highlighted in blue that are found in very young patients. So that means that there's a clinical heterogeneity, not only at the level of the symptoms, but the level of age of onset. So I think that if you have a member family with an LHON mutation, whatever it is, I think it's really important just to then follow up immediately with when you have children, just to make sure that they don't have another variant that can be triggered during early onset. So we present always LHON and LHON plus as an adult onset disease, but there's always exceptions. But 3 variants, I think it's more than an exception. So then now I'm just going to address this mystery that we will all want to solve. Why do we have so much clinical heterogeneity, not only among patients, but among family members? So one family member can just have vision loss. Some others can just be carrier, but be asymptomatic. And some can have like extraocular symptoms. So what triggers that? So James actually brought up the notion of heteroplasmy, and I'm just going to bring out to another level. So he presented as a whole. I'm going just to say now, to make the statement that this level of heteroplasmy varies from cell type to cell type. So you can be homoplasmic in one cell. For example, I'm going to say the retinal ganglion cells, because those are the ones in question leading to vision loss. But it doesn't matter. You can have also a significant heteroplasmic level in cardiac muscle cells. And hence, that's why maybe when you mentioned in your email that you have like several patients that actually pass away from cardiac problems. So that's why we want to expand and the notion that it's the spectrum, and that the patient should not, a patient with LHON mutation should not just be treated just for the vision loss, but maybe other symptoms that can appear later in life. And the difficulty here is just to find whether it's just due to common aging, like we all going through, or it is due to something else. So it's a lot of heterogeneity among the heteroplasmic level within cells. So I'm going from one extreme, the left, to the right. The extreme on the left is like a homoplasmic, and everything is fine. All the mitochondria are wild type. So that means that you produce normal ATP. If you swing totally on the right, then that means that all the mitochondria carry the mutation in question, and they are homoplasmic. And that's when usually there's symptoms. And then you have all the in-between. And that's in between we're also interested, because this is maybe responsible for this intense clinical heterogeneity among patients. So if you have, there's a threshold here, and we think that above a threshold, like I'm just putting it arbitrary at 70%, then the cells in question will not make enough ATP, and then you have a dysfunction. So if you're below that threshold, you'll be maybe limping, the cells will not be super duper performant, but it will not translate into clinical symptoms. So that's why, I mean, you have so much heterogeneity. But then there must be something more, because I mean, you know, you're not, you can see that because I brought the notion that the mitochondria needs the helper, the nuclear genome to function, then you can really conceive that maybe the nuclear genome will actually modulate the penetrance of that variant in question, that pathogenic variant, meaning it can either exacerbate the penetrance or have no impact. And I think that's why, I mean, we need just to go beyond sequencing the mitochondrial genome, but doing whole exome sequencing and eventually whole genome sequencing, because I really believe that's where the secrets lie. So, and of course, like I said, then while you have some carriers that are asymptomatic, so maybe they don't have that modulator in a nuclear genome that would just tweak the penetrance, make it worse. Or maybe I could think kind of a positive manner as well. I could think that maybe the carriers have something, a modifier that would dampen the penetrance. You know, you can either exacerbate or really decrease the penetrance. And I think that's where the questions will be there. So right now, our strategy at the Children's National Medical Center and at the George Washington University School of Medicine, where I am, we decided just to build a strategy toward precision medicine, just to address this clinical and genetic heterogeneity, because we realize that one size fits all is not going to work. Because you either then yes or no, it's kind of a binary decision. And then so many, so it's fine when it works for you, but then when it doesn't work for you, what do you do? What's available to you? So how come we haven't been there? How come we haven't achieved precision medicine? It's because there's so many unknown in terms of the pathogenic mechanism. And that once we crack that mystery, then it's just going to open the so many avenues to design, to do custom design therapies for patients. So what do I mean by precision medicine? I mean that digging into the mitochondrial energy signature and the pathogenic mechanism of each patient, and see, right now you classify vision loss, no vision loss. Do you have something more or not? So it's a very broad and general categories. But we want to say, okay, among the vision loss, are you all equal or is not? What's the difference? And for that, we need just to find what is the mitochondrial energy signature for each patient. So what is your defect? Okay, we all understood that there's less ATP, but due to what? That's a question we want to answer. So, because right now the design of all the clinical trials is for the average patient, and also one more important point for the precision medicine, based on the discussion that we had on the previous session on clinical trial. And what I heard is a lot of complaints from the patient and the patient's families as well, that say, you know, then how do you know which clinical trial is the best for you, for your loved one? It's a tough question to address, because if we don't know the mechanism behind it, specifically that patient, then it's a wild guess. So then you cannot jump from one clinical trial to another one. There's also a heavy cost associated with it. So, and I think I heard loud and clear those are not complaints, those are legitimate concerns expressed by the families. So I think it would be really a win-win situation for the patient if we will go beyond the average patient strategy and really tailor it to the patient. So what can we do here? What do we do in the lab and in collaboration with Dr. Goldman? Because it's a very symbiotic collaboration I have with Dr. Goldman. Dr. Goldman, she not only she comes up with a diagnosis, but also she does the skin biopsies on patients. And then we bring it into the lab, and then we derive the patient cells, And then we test them. We do all kind of tests just to see where the deficits reside. And then we have like a battery of therapeutical compound that we can test on each patient cell and see whether the patient cells respond to those. So I understand it's in vitro, it's not within the body, but it gives you a clue whether this patient will be a good subject for a specific clinical trial targeted with a targeted drug. And we also do something else for metabolomics and proteomic analysis. And that's where we combine all the protein that you make in the mitochondria that are specific to you and see whether there are deficits in there. So when we put together that, then we can stratify the patient to make more meaningful clinical trial. So that's our strategy here. So this is a little cartoon to explain what we are doing in terms of diagnosis in the lab. We go beyond the genetic diagnosis. We also do the energy diagnosis. So in other words, we're putting the mitochondria a little treadmill. So on the left, you have the treadmill that is going on that mitochondria on the treadmill with really low speed. So it's not doesn't need much. I mean, just like slugging along. And then say, okay, fun is over. Now we're going to make you work. So chemically, we kind of force, it's like we increase the speed dial, and then we force the mitochondria just to run really fast. And then we realize that most patients with LHON plus or LHON MS have like a deficit in the reserve energy. So that means that their mitochondria are not able just to sustain this really rapid speed on a treadmill. And that leads to different symptoms such as chronic fatigue, exercise intolerance, mental exhaustion, muscle weakness, neurological symptoms, and gastrointestinal dysmotility. So, and it's essentially due to the lack of reserve energy. So then, I mean, just to end up on a positive note, because I mean, I think that we need just to bring it to one step higher, is that then how do we diminish that deficit? How can we give more reserve energy to to those mitochondria, such that they might not be as performant as a patient or someone with no mutation? But you know, when I said that if we can dial up or dial down the heteroplasmy, then that's going to have a big impact on clinical symptoms. So it would dampen the clinical symptoms. It would just improve the quality of life and so forth. Because I think sometimes it's lost that there's not just loss of vision. There's a lot more symptoms that are associated with those patients, and fund those patients, and the others are not really addressed. So that's what we're doing in the lab. We are just trying to make sure that then your mitochondria can run on the high-speed treadmill. And of course, I want to thank Lisa and Martine, who is in the audience here, that has been the lead research scientist, and she has been cultivating those cells. I think she must dream at night about all your cells. So thank you so much. And it seems I'm just going to have very few time for questions because I have my plane to catch. I'll be very, like I said to Lisa, I'll be very happy to do a Zoom session with you just to go over other questions or other concepts that were not clear. And in the meantime, you can always visit our website on that hyperlink. Thank you.
Thank you so much. I'm going to let you catch your plane, and thank you so much. I will say that I posted last night, you were interviewed by the UMDF team, and I posted into our LHON community that you would be speaking today. And somebody said, oh, I've got my cell skin biopsy is culturing in her lab. Please, I feel like I'm hanging out with her at work every day, and she is an absolute gem. And so just from the patient perspective, people, thank you so much for being a gem and for all the work you're doing on our behalf. And again, thank you so much. Okay.
As I said earlier, I've enjoyed exploring different topics within the world of LHON to help create accessible content for our community. But the topic of migraine holds a particular interest for me. While I don't have an LHON mutation, I do experience migraine myself. Migraine is quite common in the general population, so it's not surprising that I myself or anyone with LHON experiences it. But migraine is so much more than a headache. You may notice that I'm saying migraine and not a migraine. And that's because it's actually considered a neurological disease by many professionals that can even have a genetic component to it. Given that both LHON and migraine are neurological diseases and that many in our community suffer from migraine, as we were just hearing from Dr. Chiarmorello and Dr. Grotman, it seemed an important topic to educate our community on. So first, I just want to describe what a migraine attack actually looks like for many people. The Mayo Clinic describes it as a headache that can cause severe throbbing pain or a pulsing sensation, usually on one side of the head. It's often accompanied by other symptoms such as nausea, vomiting, extreme sensitivity to light and sound. And migraine attacks can last for hours to days, and the pain can be so severe that it interferes with your daily activities. So is there a connection to LHON? So most LHON research in clinical care is focused more on vision loss and is done by neuro-ophthalmologists who may not be investigating non-vision symptoms necessarily. And a connection has not been fully proven in any way, but a very high percentage of those with an LHON mutation, whether they're affected or not with vision loss, experience migraine. For example, in an unpublished study of LHON in women, 55% reported migraine. But what does the research say? LHON, of course, is a mitochondrial disease, and there is some research that indicates there's a connection between mitochondrial disease and migraine. Most LHON investigations focus on those who are affected, so about 75% are male, overlooking female LHON carriers who don't have vision loss, but who often report migraine. So while there are some parallels in the literature on mito disease and migraine, it's been largely left out of LHON research. So of course, more research is needed to explore the relationship between mitochondrial disease and migraine more broadly, and more specifically, LHON and migraine. So what are the next steps for assessing and understanding LHON and migraine? Of course, we need to continue to investigate this relationship, as I said before, but we really need to educate. And that education needs to be evolving to always reflect the best available evidence at the time, as is true, of course, for all of our educational materials that we're developing. In terms of investigating, we want to see, is there a connection between LHON and migraine, or even more broadly, migraine and mitochondrial disease? There are hundreds of people in the LHON community who could be studied to explore a variety of hypotheses about migraine and LHON. And the exploration of beneficial and potentially harmful treatments for those with an LHON mutation would be very valuable to investigate. On the education side of things, of course, we want to let people know that there may be a connection between migraine and LHON, or migraine and mitochondrial disease. It's really important for folks to be aware that it's always beneficial to see a mitochondrial disease specialist in general, but even specifically if you're experiencing migraine. There are also professionals out there who specialize in headache, which can be broader than migraine. It can include other types of headaches, and those individuals might be really helpful in helping you manage your care. There are several prescription drug, non-prescription drug, and non-drug treatment options available to prevent and treat migraine. But always, we recommend that when meeting with any medical provider, whether you're affected with vision loss or not, it's really important to remind them that LHON is a mitochondrial disease, kind of like what James and Elizabeth were speaking to earlier. So much like the Charles Bonnet syndrome handout that we were talking about before, we have a new LHON and migraine handout. And as I said, we need to constantly evolve our educational materials to reflect any new learnings. But for now, we don't know a whole lot about the two. So what we do know, we've synthesized together in this handout. It's available in the UMDF app in the family syllabus section called LHON Saturday Science Migraine. We will also be making that available on LHON.org following the conference. There are so many resources out there to support you. You can find a mitochondrial disease specialist near you, going to mitonetwork.org. You can look for a headache specialist near you. The American Migraine Foundation has wonderful information about new treatments. You can also follow the latest in migraine research every year by attending the Migraine World Summit. It's a wonderful virtual event that I've attended a few times and learned so many great things. I wouldn't be surprised if they have talked about migraine and mitochondrial disease before or will in the future. And there are a variety of other resources, all of the ones I've mentioned and others included in that handout. So I'll leave with a quote here. This is from Dr. Matthew Robbins, who presented at the Migraine World Summit this year. And he said, "You have a legitimate inherited neurological disease." And I think it's a great quote because it applies not only to LHON, but also to migraine.
My name's Melinda Marsh, and I'm reintroducing myself from yesterday. We all went around and said who we were and how we were a part of this LHON community. And I'm going to start tonight or today with a little bit about my journey. So as you already know, I have 3 sons, 1 affected and 2 who are carriers, just as I am. Xavier was diagnosed in September. And I would say, arguably, everyone in this room has a connection to Lissa, and that's one of the reasons that we're all in this room. And I was spiraling in September. What do you mean I have this mitochondrial DNA mutation? What on earth are you talking about? What do you mean, my son, there is no cure? What? And Lissa reached out after my request to join the Facebook group and said, "Hey, can we meet offline? I'd love to talk to you." She gave me 3 hours of her Sunday, having never met me and never experienced. We weren't friends. We didn't even know each other's names prior to that conversation. And she talked me off that ledge. She brought me to a point of, I'm so grateful to have had this LHON community that Lissa worked so hard to establish, to have that network to lean on and to figure out supports for Xavier and for the rest of my family. And from there, how do I take that gratitude and pay it forward? And that's why I'm here today. And I'd like to introduce my husband, Chris, as well.
You held that together much better than when you practiced it.
I did.
This is an emotional journey without a doubt. And with that whirlwind of emotions that I think Melinda just described, we as 2 type A individuals went immediately into action. And upon diagnosis, we were fortunate to have some friends in the life science community that were able to get us a diagnosis in about 48 hours, where we know in so many cases it takes much, much longer. And in the ensuing weeks and months, we're sure there was a fix out there. So we had Xavier enrolled as a patient in Los Angeles and Miami and in London, and just knew we needed to turn over that next rock we just hadn't found. Sound familiar? And in so many cases, while that fix is perhaps elusive still, what we've learned is what an incredible network of talented people exists in the research community and need your help, need our help to actually bring down some of the walls and some of the obstacles. And so that's what we wanted to talk with you about, is how to actually launch a data collection program for LHON that we think can do exactly that.
So prior to September, if somebody had said the words medical registry to me in that order, I would have looked at them blankly and had no idea what they were speaking about. And just in case any of you are in the same boat that I was in, a data collection program or a registry, medical registry, this is a systematic collection of that standardized data, our data as LHON community individuals. It captures that information about each rare disease, including LHON communities' symptoms, whether it's vision loss and in addition to vision loss. And then it facilitates getting that information in the hands of clinicians, biopharma, medical researchers, and so on, so that that can be moved forward towards clinical trials and studies to develop those effective treatments. And I'll add that as Chris and I were hunting for the elusive cure, and we met all these clinicians, everyone said they then introduced us to biopharma partners that we then followed up with and became friends with. And as we continued to meet individuals in the LHON space on the medical profession side, what kept being, how would you say, honed in on and hammered into our brains was, you have this amazing community, this support network. Her name is Lissa Poinsano. What we need from you as the LHON community is a quality medical registry so we can move forward with these great ideas to possible and very probable treatments in the near future.
So we recognize that a lot of data is out there. And so I think it's really important just to understand what are we trying to accomplish incrementally. Across the board, I think it's fair to say that data exists in silos. You know, when we flew down to Miami, we had a chance to meet with the Bascom Palmer folks. And those of you with 11778 certainly would be familiar with some of their attempts and their trials. But it wouldn't surprise you that that data isn't available to other research institutions, much less like the learnings behind it to actually build on in an effective way, not nearly as effective as it should be. And so the idea of building this this new sort of data collection platform is really to create something that is accessible, but also representative of all of what's represented in this room. And I think the last 2 hours of conversations and symptoms that go way beyond just simply a single diagnosis. And so as Melinda and Lissa and I went out and spoke with various data collection platforms that are out there, you know, there were a few very core principles we were looking for. And we think we found a platform that can do exactly that in terms of making things more accessible, more structured, and more comprehensively representative of our community.
I'd love to talk about the beta testing. So we here up on this stage aren't going to ask you to do something that we haven't already done ourselves and had some proof in the pudding that it works. So when all of these medical professionals said to me as I was meeting through Zoom in person and so on, that this medical registry is a key next step. I said, okay, fine. I'll register everywhere under the sun. I don't care if it's in English, I will do it. And so I did just that. And I found a lot of gaps in those medical registries that I went forward and registered Xavier as an affected member, myself as a carrier, if they would take my information. And I kept looking and I came finally to RareX, and they have a very different approach than simply a static, potentially upload your genetic report, potentially don't. Maybe they have LHON in the dropdown, maybe they don't, etc. And what RareX did that differentiated themselves was they have this head to toe approach. And I reached out to Lissa and said, I think I found something unique. And so then Lissa and I went in and tested it ourselves. And they ask everything from your vision loss to your migraines, to muscles, to your heart, to everything from the top of your head to the bottom of your toes. And what that does is it gives us in the LHON community a whole picture for those researchers to access, to understand our DNA, mitochondrial DNA mutation. And so we've gone to a beta test only as of last week with a very small subset of LHON community participants, just to confirm that what I think is an awesome opportunity ends up being an awesome opportunity for others within our community. And assuming that their response is also positive, we want to then roll this out to the greater LHON community, not only here in the United States, but internationally, because as has been shared in many different sessions here with UMDF, as well as in this room, LHON doesn't understand borders. And so there's no reason that we should keep this proprietary to our borders as well. So the next slide are about our goals. Our goals here are to create a robust database. We need a large number of participants to create a quality set of information that can be used by those researchers. In addition, this has the potential to reduce the time it takes to study those new medicines and to promote possibly the development of new assistive devices.
I'll touch on just a little bit on the collaboration point. And those of you who were here yesterday heard from Carmen Trupik as she described a little bit of what RareX is doing and how important collaboration is. The key things that I would mention on simplifying this slide down is that we're talking about what you're experiencing, what our families are experiencing, and an aggregate picture of symptoms, not just a specific diagnosis. The collaboration that can come from that really fosters a cross-disease, cross-diagnosis type of approach to really understanding what's going on. And I think a really important piece is that they're not trying to replace other studies. One of the key elements that Lisa and Melinda and I emphasized when talking with RareX was the importance of partnership with MitoShare, so that we're not creating things that are redundant. What we're actually doing is creating multiple access points and abilities to query the underlying data and actually make it relevant to the researchers. So why is this going to help us with researchers? Well, at the end of the day, if we can reduce some of the obstacles that researchers face, we become a greater priority for them. Let's just be very blunt about it. They have lots of options where to spend time and energy. If we can actually reduce obstacles, we become a priority. And in addition to that, there's some really amazing ways to harness the data science that's out there. Again, if we start to think horizontally, not just by vertical or by silo. And so one of the most fascinating parts is not just what RareX has today, but where they're going. They've created a partnership with the Broad Institute. If you're familiar with that, it's a joint venture between MIT and Harvard data scientists to actually look at it from an algorithmic data standpoint, to turn data into actual information that can target efforts going forward. And that we think has benefits not only to researchers, but to all of us as a community.
So we're going to move on to us as a community. What are our benefits to being a part of this DCP, data collection program? The obvious main benefit is the access to those clinical trials that any one of us may be potentially appropriate to participate in. In addition, though, this also has the possibility of speeding up research and drug development, as Chris just mentioned, and it will also give us, as we have a greater number of participants enter their information and keep it updated as your symptoms change or your vision improves, for example, or you experience a migraine, et cetera, and the list goes on. The ability to receive updates on those disease commonalities that we may experience within our LHON community to better understand ourselves, what we share in common. So the last stage here is who can enroll. And this gets down to the nitty gritty here. Obviously, anyone affected with LHON that is traditionally understood as anyone experiencing vision loss. In addition, though, just as the genetic counselor was mentioning earlier today, anyone who is known or presumed to have an LHON mutation, even if we don't have vision loss, commonly referred to as LHON carriers, such as myself, such as my sons, Danny and Sean, we all carry that LHON mutation, and we are important to that overall research. So as we roll this out, it will be very important for not just those considered to be in that affected category, but those in the carrier status to also participate. Anyone with a diagnosis of LHON plus, and lastly, anyone who is deceased but known or presumed to carry that LHON mutation. So my mother, who's passed away, would be appropriate to enter. She did not lose her vision, but she had other what would be considered, as I'm now learning, LHON plus symptoms. And at the time I had no idea. I didn't even know LHON existed. So thank you so much for your time today. I really appreciate all that you've given us as a community. We are so lucky to be here today and to have this opportunity to make these connections and these friends. And we want to work together to see what may be possible in the very near future.